What if I told you that everything in this life is an imitation of nature? What if I told you that the buildings you see, the houses you see, the roads, the vehicles, and every other thing you can imagine actually emanates from you? Quite surprising, right? Journey with me as we uncover the origin of everything in this our reality, which is self. In this video, we'll be looking at cytology. Now, what is cytology? Cytology basically means the study of self. So, as you can see here, cyto is an anatomical name or a medical terminology we use in referring to self. While logy means to study. So, if you were to combine this, it means study of self. Now, before I continue, what exactly is self? I'm not talking about self on the basis of definition, but understanding exactly what a cell is. Now, if you were to look at this structure, we realize that this structure was made out of blocks. In other words, it is the repeated arrangement of blocks that brings about this building. So likewise, in the human body, we have this repeated arrangement of cells which form who we are or form us as living organisms. Now, if we were to remove all the blocks they use in building this place, you agree with me that this building will collapse. Likewise, this shows that cell is also the fundamental, basic and structural unit of life. If we were to remove cells from living organisms, it means that that thing does not have life. So what cell basically means in this context, it is the smallest unit in which life is found or in which life can be exhibited. But then the technical definition is cell is defined as the basic, fundamental, structural and functional unit of life. So that is that about the definition of cell. The next thing I would like to talk about is the cell theory. Now, before talking about the cell theory, I would like to refer back to how this cell as a scientific discipline came into play. Now, the idea of cell or the publishment on cell was first done by Robert Hooke in the year 1665. So, Robert Hooke was the first person to make an observation or to publish articles that were related to cell, and he is referred to as the father of cell. So his name is Robert Hooke, and the year is 1665. So in 1665, Robert Hooke made a publication, and he was or he is still currently regarded as the father of cell. That is that about how cell began. And now moving on to cell theory. Now, the cell theory was proposed in the year 1838 by Matthias Jacob Schleiden and Theodor Schwann. Now, these were the two scientists that proposed the cell theory. Now, in the early 1800s, both Matthias Jacob Schleiden and Theodor Schwann made an observation. First, I would like to state that Matthias Jacob Schleiden was a botanist, and by botanist, I mean he was somebody that studied plants, while Theodor Schwann was a zoologist, meaning he was somebody that studied animals. Now, both Matthias Jacob Schleiden and Theodor Schwann were busy experimenting on both plants and animals. And the result they had was that both plants and animals were made up of cells. Now, this brings them to the conclusion that all living things are made up of cells, which is one of the cell theory. So, one among the cell theory is that all living things are made up of cells. In other words, for anything to be termed living, it has to be made up of cells. Another aspect of the cell theory is the fact that cell is the basic, fundamental, structural, and functional unit of life. In other words, life in its structure is found in cell, and also the functioning of life is exhibited at the cellular level. And if you were to go beyond cell, there is no life. Another important figure that contributed to this cell theory is Rudolf Virchow. Rudolf Virchow. Now, Rudolf Virchow made a statement and it still added or it contributed to the cell theory. And that statement is that all living things arise from pre-existing cells. In other words, the way that cells are formed is that they emanate from an already existing cell. And that is his contribution to the cell theory. So basically, the cell theory is trying to enlighten us on first that trying to enlighten us on how everything that is living is actually made up of cell, and also how this cell is the basic fundamental structural and also the functional unit of life, and again how cells arise from pre-existing cells. So that is basically that about the cell theory. And one thing I want to talk about now, which is the fascinating aspect of cell, is why is it that cells are microscopic? In other words, why can't I just see cells with my naked eye? Now, you might think that is how it is, but no, in science we are trying to understand mechanisms or why things happen the way they do. So there are limitations to a cell size, or there are limitations to how big the cell can grow. And this is what we'll be looking at shortly. We want to look at why cells are limited to a certain size. Now, there will be an inclusion of mathematics in this. Hope you enjoy it. But then, let's look at what a cell is and how cells perform their function. Now, the way that we obtain energy, or the way that we gain energy in the form of ATP, is as a result of what we call cellular respiration. And cellular respiration is basically the name of the process whereby organic molecules such as sugar or glucose is broken apart in the presence of oxygen and releasing energy in the form of ATP. So the way that cells operate is that sugars and oxygen has to get into the cell in order to manufacture this high energy nucleotide, which is the ATP, and then at the same time waste product that is being formed within the cell has to get out of the cell in form of carbon dioxide. So likewise, we have vitamins and minerals. Vitamins and minerals, which are also referred to as coenzymes. What they do is that they have to get into the cell fast enough in order to assist enzymes in carrying out um, biochemical processes, and if they are not able to get into the cell fast enough, then those enzymes may not be able to carry out those biochemical processes. And another thing again is that water also has to get into the cell and also get out of the cell. So using this pattern, it means that if this glucose or oxygen is not able to get into the cell fast enough, then the cell will die because this energy that is needed to power up the cell will not be made, which is ATP. And likewise, if after this glucose and oxygen gain access into the cell, and then the waste product formed, which is carbon dioxide, is not able to get out of the cell fast enough, then the accumulation or the buildup of this CO2 will cause the cell to die. So basically, what it means is that in the operations of cell, they are working within a time range. In other words, there is a time whereby if the nutrients cannot get into the cell, then the cell will die because ATP is not being produced. Likewise, if these accumulations of waste products such as CO2 accumulates in the cell, then it will cause the cell to die. What this means is that cells are working on time, or the activities of cell is guided by time. And I want to use this scenario. Let's say, for instance, I was to have something like this, a circle like this, and comparing to this particular one, right? It will take me lesser time to move from this region to this region compared to this one. So if it were to take me about 1 second to move from here to here, it will take me about twice or thrice of that time to move from here to here, right? So this is the same analogy on how cell operates. They have to be a fast exchange of both nutrients and waste products. Now, that is just a picture, but then let's go to the mathematics aspect, which is alright. Now, this membrane or the outer covering of the cell is what we refer to as the surface area of the cell. While what is inside of the cell is what we call the component or the internal volume of the cell. If you look at the formula I have here, they look alike. Now let's look for the similarities. We have pi here, we have pi here. So we are no longer concerned with pi. We have 4 here and 4/3 here, which the difference is not that complicated. So let's remove these two. Now you realize that the difference lies in the radius, which is in the surface area, the radius is squared. While when it comes to the internal volume of the cell, this radius is cubed. So basically, in explaining the limitation to the growth of a cell, what we are considering is the radius of the cell, which is the surface area, and then the cube of the radius, which is the internal volume. Internal volume. Now, this is the difference that lies within the cell. So let me use this as a case study. Suppose this is the radius of a cell, and let's say it is 1, and I have another bigger cell somewhere here. The radius is 2, right? If we were to calculate the surface area of this cell, it will be the radius, which is 1 squared, and 1 squared is 1 times 1 is still 1. So for this cell, we are having the radius or the surface area of 1, and also the internal volume is radius cube, which is 1 times 1 times 1. We are also having internal volume to be equals to 1. Now, there is a significant difference when it comes to this. It means that for this particular cell, both the internal volume, that is the content of the cell, and also the surface area are the same. So it is for the movement of nutrients fast enough, and also for the removal of waste products across the cell membrane. Now for this particular one, we have 2 to be our radius. And from the formula, if we're calculating for the surface area, which is this area of the cell, the radius is squared. Now, the radius is 2, and if you are to get or to look for the surface area, it will be 2 squared. And 2 squared means 2 times 2, which is equals to 4. Now, for the internal volume, which is the content of this cell, what is inside of this cell, it will be this radius cubed. In other words, it will be radius times radius times radius, which is 2 times 2 times 2. 2 times 2, 4. 4 times 2, is 8. So you can see the relationship between the surface area and the internal volume. Now, the surface area is increasing at a slower rate compared to the internal volume. In other words, the internal volume is growing at a very faster rate compared to the surface area. And since the internal volume, which is the content of the cell, is growing at a faster rate compared to the surface area, it means that all these molecules that are to get into the cell and perform their action fast enough will not be able to do that because it takes a longer time for these activities to occur as a result of increase in the internal volume of the cell, and that is what causes cells to die. So if a cell has attained a certain growth or a certain level, it either do one of these two things. It either stop like that and stop growing, or the cells divide into two small cells. And the way that we grow is as a result of the divisions of these cells. So that is basically that about the limitations or the reasons why cells remain a certain size. It all boils down to the relationship between the surface area and what is inside or the internal volume of that cell. So that is that about the limitation to cells growth. Now the next thing I would like us to look at is types of cells. Another aspect we want to look at is the types of cells. Now there are basically two types of cells, which are the prokaryotic cells and eukaryotic cells. Now the name pro, first of all I would like to define the terminology. Pro simply means before, right? It means before or referring to primitive. There are cells, while karyote refers to nucleus. So there are cells that evolved before there was a nucleus. In other words, there are cells that lack nucleus. While in the eukaryotic cell, U is a Latin root for true. So it means they are true nucleated cells. And as you can see, this is a nucleus. First of all, what is a nucleus? If you can recall in chemistry, we say the center of an atom is referred to as a nucleus. So nucleus simply is referring to the center, right? And as you can see, in this center lies the genetic material of the eukaryotic cell. While in this is the genetic material embedded in the cytoplasmic fluid. So the prokaryotic cells are cells that lack true nucleus and also lack cells organelles. In other words, they are simple cells. They lack organelles that say or carry out cellular activities, and also their DNA material is embedded within the cytoplasm. So there is no covering or a special membrane that segregates the DNA from the cytoplasmic fluid. Now, coming to eukaryotic cells, which are the true nucleated cells, is that as you can see, they have a membrane just like this. They have what we call nucleus, and it is in this nucleus that the genetic material is embedded. And another thing to note about them is that they also possess organelles. So there are different organelles in these cells that account or that are responsible for different actions of the cell. For example, we have the ribosomes, the mitochondria, the Golgi apparatus, lysosomes, and so on. So all these organelles, we'll look into them after this aspect, but then all these organelles are responsible for carrying out specific function within the cell. While on the other hand, which is the prokaryotic, they do not possess all these organelles. And to add to it, they possess what we call a cell wall. So there is a cell wall. In the case of a bacteria, let me be specific, bacteria, they possess a cell wall, then a cell membrane. So in similarities, you can see that we have a cell membrane, cell membrane. But again, in the case of plants, plants also possess cell wall. And that is that about the types of cells. So in prokaryotic, we say there are cells that do not or that lack a true nucleus and other cells organelles. While when it comes to eukaryotic, we're talking about cells that have true nucleus and also possess organelles that undergo some specific actions within the cell. So that's basically that about the two type of cells.
The last class we had, we discussed about the types of cells. We also discussed about the limitation to why cells grow. Now, in this video, we'll be discussing about organelles of cells. And the first organelle of cell we have is the cell membrane. Now, structurally, the cell membrane is made up of bilayer. So the cell membrane is made up of bilayer, and by that, bilayer of phospholipids. So the composition of the cell membrane is a bilayer of phospholipid molecule. Now, this is the structure of a phospholipid molecule. And not only is it made up of bilayer phospholipid molecule, but there are proteins embedded in the cell membrane. Alright, so this is a typical diagram of a cell, and we want to enlarge this section of the cell. This section of the cell, we want to make it in a bigger form. So this is what we have here. Now, just like the way it is here, this is the cytoplasmic fluid. So we assume this side to be the cytoplasmic fluid or the cytoplasm. The outer part of the cell, which is this part of the cell, is called the tissue fluid. Now, an interesting fact about cell is that for every cell or all living cells are surrounded by fluid in order to be alive. Now, all of our cells as humans are surrounded by fluid in order to be alive, or the surrounding of this fluid help bathe these cells in order to keep them alive. Now, if that statement is true, which of course it is, why then is our skin not surrounded by fluid? Or is it that our skin or the sweat we bring out from our skin are the fluid that bathes our cells and then keep them alive? No. The answer to that question is that one of the characteristics of the cells of our skin is that they are composed of what we call epithelial cells. And epithelial cells, one of their characteristics is that they lack blood vessels. And aside that, whenever the skin comes in contact with air, the cells automatically die. So that is why there is no nourishment or no fluid surrounding the epithelial cells or the dermis of the skin, the epidermis. Now, another thing worth saying here is that we've established the fact that cells, first of all, are surrounded by fluid in order to be alive. So the next thing I would like us to look into is the composition of this cell membrane. So this is just an enlargement of this section. So we have made an enlargement of this section, and that is what we are having here. So let's discuss briefly on what all these things are. Now, we said, first of all, it is made up of bilayer of phospholipid molecule, right? Now, not only is it made up of phospholipid molecule, but also with proteins, embedded proteins. So, as you can see in this diagram, this is the bilayer, this layer 1, and the other side is layer 2. So all these things you are seeing, these bigger molecules, are proteins, and I will discuss all this briefly. So let's first explain what this bilayer means. Now, if you look at this structure, it kind of look like a balloon with strings attached to it. This is how the phospholipid molecule is. And when we say bilayer, it means there is a part that is facing the tissue fluid and another part of the same phospholipid molecule facing the cytoplasmic fluid. So as I said earlier, it looks like a balloon with two strings attached to it, and the balloon part of the molecule, which is the PO4 or the phosphate aspect of the molecule, is said to be hydrophilic. Now, since we said all living things are surrounded by water, it means that this tissue fluid is fluid in nature or watery in nature. And since we say this is hydrophilic, meaning it likes water, then you can see that the balloon aspect is situated within the fluid or the tissue fluid region. Likewise, it is the balloon aspect that is situated inside of the cytoplasmic region. So this part of the molecule likes water. That is why it's found in the tissue fluid aspect and also in the cytoplasmic fluid aspect, which are all these balloon shaped molecules. Now, the next thing is the fatty acid or the lipid aspect of the molecule. Now, these fatty acids are said to be hydrophobic. And hydro means water, phobic means hate. So they hate water. A common example I will give is, let's say for instance you have oil, regardless of which type it is, and then mix it with water. Now it is very possible that the result obtained will be that the water or the oil itself will suspend. The oil will separate from the water, and oil or fat and water do not mix. That is why it separates itself. So this is also the arrangement. Because this part of the molecule, which is the fatty acid part, hates water, that is why it is situated in between. So if you think of the cell membrane, it's more or less like sandwich, or it could be a burger. Why? Because this part is more or less like the bread part. This other side also the same thing. Then in between lies all the creamy fillings that are in it. So that is that about what the cell membrane is. Now, the next thing we'll be talking about are the roles of proteins in the cell membrane. Now, there are different functions of protein that are embedded in the cell membrane. And one of the roles of this protein are, first we have the ion channels. So as you can see clearly, this part or this extension, this tube-like extension that cuts across the cell membrane, that is forming a tube-like pattern, is what we call ion channels. Now, the ion channels, as the name suggests, are passageway or are mediums whereby ions can get into the cell or get out of the cell. So ions, first of all, are defined as chemically charged atoms. Ions, example of ions are, we have calcium ions, potassium ions, we have chloride ions. All these are ions. They are atoms, but they are chemically charged. Now, the protein or the ion channel, which is a protein, its function is that it helps in the transportation of all these ions into the cell, and also some can get out of the cell via this medium. Now, it is worthy of note that not all or not a single ion channel is capable of transporting all ions. In other words, every ion has a specific ion channel. So there are different sodium ion, there are different potassium ion, there are different calcium ion channels. So each of them are independent of their ion channels. Now, what is the importance of these ions? Well, ions help in coordinating the electrical activities of our body. For example, the actions of our heart or the actions of our nervous system are electrically inclined. In other words, these are ions that help in controlling those activities or those actions electrical actions of the body, and that is that about the ion channel. So the ion channel, in summary, is a protein and it aids the movement of ions in and out of the cell, and it is specific to which ion gets into it. So each of them have various separate ion channels that are different in terms of their transportation. And that is that about the ion channels. Now the next protein which we would like to look at the role of that protein in the cell membrane is enzymes. Enzymes. Enzymes are proteins that catalyze biochemical reactions. Now let me give you a very common example of enzymes. Now growing up, I used to have friends and we normally went out to play with each other and stuff like that. Now on rare occasions, we normally fight among ourselves. And just so you know, I have friends that are not really involved in our fight, but they keep on gingering us or keep on encouraging us to fight. Right? So if we were to start fighting with a friend of mine, what they do is they come there and say, go, you can do this, beat him, stuff like that. That is their role. At the end of the fight, they will not suffer the pain, but they alter or they cause, they fasten the rate of our fighting. So that is the same idea about enzymes. Enzymes are chemically proteins that catalyze biochemical reaction without them being in the product. So all they do is that they alter or they fasten the rate of chemical reaction without being involved in the reaction. So there are enzymes that are located within the cytoplasmic aspect. There are enzymes that are located in the tissue fluid aspect, and that is that about the enzymes protein. The next thing I would like us to look about is what we call transport proteins. They are called transport proteins or carrier proteins. Now, what this transport protein or carrier protein does is that they aid in the transfer of larger molecules such as carbohydrates and also amino acids. Now, first of all, we made mention that the ion channel is specific to which ion pass through it. Likewise, enzymes are specific to which biochemical reaction they cause. Now, even this thing called transport proteins are specific upon which molecule pass through them. So in other words, there are different transport proteins for molecules such as sugar. There is a different transport protein for amino acids and so on. So they are specific upon which chemical or which compound gets into them. Now, this idea is, to me, I believe, is where the origin of vehicle came into place. Why? Because the idea that you need to pick up something and move it actually began from here, within the cellular level. So what they do is that after receiving those molecules, they pull them into the cell so as to fulfill their purpose of being in the cell. So you can see that these are responsible for the transfer of larger molecules such as carbohydrates and amino acids. And again, we made mention that they are specific to which molecule is getting into them. Now, the reason why this transport protein is different from this is because transport protein carries larger molecules. For example, glucose, C6H12O6, that is about 24 atoms big. And you cannot compare the part that is about 24 times bigger than this. So it will require a separate or bigger medium in which it can be transported into the cell. And that is where the role of transport protein comes into play. The next thing I would like us to look about, which is very fascinating, is what we call receptor. Now, these are called receptor sites. The receptor site, as the name suggests, are sites whereby molecules or certain things are being received. So, an example of chemicals or example of things that the receptor site protein receive for the cells are hormones, hormones, neurotransmitters, and drugs. Now, first of all, what are hormones? We said hormones are chemicals that are secreted into the bloodstream and then causes a change in a cell upon reaching that cell. First of all, one of the common hormones which I will make reference to is let's talk about progesterone or estrogen, for instance. Estrogen, which is the female sex hormone, affects only the cells of the female body that is responsible, especially her reproductive cells, her uterus. That is what the estrogen affects. Now, the reason why estrogen affects the uterus and not the heart cell is because there is only a receptor site that can receive this hormone in the cells of the lady or in the uterine cells of the lady. So to clarify a little bit, what I mean is that receptor sites are simply like a channel that receives certain information. Now, because our cells are composed of different, in fact, a cell can have as low as 2 receptor sites. Some can have 20 to 30 receptor sites. But then it is on the particular cells that those receptor sites for hormone are there. That is when these hormones attach. So the reason why the hormone estrogen does not affect the human heart or does not really attach to the human heart or a lady's brain is because it is only meant to affect, or the only receiving site for it is found in the cells of the uterus of the woman. And that is that about receptor site protein. Second thing I would like to make mention of is neurotransmitters. What are neurotransmitters? If you are familiar with hormones such as estrogen, testosterone, somatotropin, oxytocin, etc., if you are familiar with hormones such as this, now neurotransmitters are chemicals released by neurons. And an example of it is dopamine, endorphins, we have serotonin, we have epinephrine, which is better said adrenaline also. All these are chemicals released by neurons. And also, the reason why certain chemicals released by these neurons have effects on certain parts of our body, especially the dopamine. The dopamine has effect only on the cells of our brain. Why? Because it is only within the cells of our brain that the receptor site for dopamine lies. So every other cell in our body, including our heart, does not have receptor site for dopamine. That is why the actions of dopamine is measured, or in fact, it is only restricted within the brain because the brain has the receptor site for it. And that is the same ideology about the neurotransmitters. Still talking on receptor side, another this thing where, or another channels where this receptor side protein help attach is what we refer to as drugs. Now, over the years, I was wondering, how come you take a drug and then it knows where to go? For example, if I'm having, let's say, heartache or headache, whatever, how does the drug know where to go? Now, the reason is the way that drugs are designed, they are designed to affect certain receptor sites. So if I'm to take a drug, let's say medication for my heart, for instance, if I'm to take it, the drug digests and is absorbed into my bloodstream and it goes everywhere around my body. Now, the actions of the drug is depending on the cells that have receptor sites for it. So now that the drug is being absorbed all over my body and transported all around my body, it will not act on any other cells unless a cells that have receiving site for that drug. So if I'm to take medication for my heart, it will be transported all around my body. But because the cells in my heart have a receiving site, like a receiving site for this drug, that is where the drug acts and cause a change in that cell. However, it is also possible that one drug can have an effect on two or three different cells receptor. For instance, the common cold drugs, if taken, you realize that it has an effect on your brain to some extent. Why? Because if you take it, despite the fact that it cures you of your running nose and whatever, it also causes drowsiness. And what is the actual reason for that? It is because these drugs for cold not only affect the cells that it was designed to, but it affects other cells that have the receptor site for it. So the actions of drug is limited to whether or not there are receptor sites for that drug. And that helps explain the receptor site protein. Now the last but not the least is what we call glycoprotein or glycoprotein or recognition site. Now this is the protein aspect of it, and as you can see, this is a sugar molecule, glyco referring to sugar, and protein to protein. So it is a combination of a protein and sugar. Now they are also called recognition site. The way that we are unique and different is that each of us have unique and different glycoprotein or recognition sites on the outer surface of our cell membrane. What that means is that the way that your white blood cells identify your cells and are able to differentiate it from foreign cells is as a result of this glycoprotein. So the glycoprotein allows your WBCs, which is the white blood cells, to identify your cells, and then to distinguish them from foreign cells. That is the function of this glycoprotein. So the reason why your white blood cells are able to detect or identify that an intruder such as bacteria is within your cell is simply because the outer membrane of the bacteria does not have the same recognition site as that of your cells. So the white blood cells goes ahead and destroy the cell. So that is that about them. And just to elaborate on how important this, even in cases of organ transplant, what we normally consider is a close match. However, recall I made mention that glycoproteins are unique, that every individual on this planet have a unique set of glycoprotein. So even if they are looking for a close match for transplant, it is not the same. And you cannot pool your white blood cells. So what they do is that after getting a close match and then transplanting an organ, they put that person on drugs. An example of that drug is prednisone. So they put that drug on that person in question, that is the person who has been transplanted a new organ, on drugs all his life. And what that drug does is that it helps to suppress or to inhibit the immune response so that the immune system not end up destroying that transplanted organ. So that is to show you how important this aspect is. And that brings us to the discussion of the cell membrane.